Three-dimensional cell culture technologies, such as organoids, boomed over the past decade. As such, precision medicine, the tailoring of disease prevention and treatment based on a patient's genes, environment, and lifestyle, is getting closer to becoming a reality for more and more diseases. First applied in the customized treatment of individual cystic fibrosis patients in 2016, organoids are now being considered for cancers as well as neurodegenerative diseases. Last year, organoids were even successfully transplanted into a patient with ulcerative colitis. Grown from either pluripotent stem cells or adult stem cells and comprising of organ-specific cell types, organoids can be efficiently established from patient-derived normal and tumor tissue samples, cryopreserved and stored in living organoid biobanks. These biobanks of different cancers and diseases can be used in optimizing drug screening. Many compounds can be screened for a specific disease, or a specific compound can be screened for many forms of a given disease. Additionally, multiple organoids derived from the same patient can be used over time to assess drug response, drug resistance, and prediction of patient outcome. Without further ado, I'm very pleased to introduce our speakers to you now. Dr. Alexander Klager serves as Director of the Institute of Molecular Oncology and Stem Cell Biology and Division Chief for Interdisciplinary Pancreatology. He is also head of the core facility for organoids, which offers services in translational cancer research to establish and cultivate organoids and to test clinically relevant substances on these cultures. His team has made decisive progress in pancreatic tissue engineering with the vision of making personalized patient care based on individualized diagnostics and therapy possible in the foreseeable future. Here, Dr. Klager will speak on the treatment response prediction and tumor evolution captured in patient-derived organoids within the context of precision medicine in pancreatic cancer. Dr. Connie Liebakken, co-founder and chief operating officer of STEM Pharm Incorporated, received a PhD in cellular and molecular biology from the University of Wisconsin and postdoctoral training in physiology and biophysics at the University of Iowa. She has extensive experience in cell biology, assay development, and advancing technologies through the product development pipeline. She has held leadership roles in operations and research and development at life science companies throughout her career, including Panvera and Life Technologies. She is currently leading STEM Pharm's research and development efforts, which focus on utilizing synthetic hydrogels to improve neural cell culture and neural organoid formation, and applying transcriptional and proteomic readouts to study neuroinflammation and neural toxicities. Please note that we pre-recorded today's presentations. However, there will be a live Q&A with the speakers following this session. You can still submit your questions by clicking the Ask a Question tab on the right, typing the question into the message box, and then clicking Submit. And now we will hear our speakers' presentations. Again, please join me live after the talks with the speakers for a Q&A session.
Good afternoon, ladies and gentlemen. It's a pleasure for me today to talk in this science webinar. And my name is Alexander Klager. I'm the director of the Institute of Molecular Oncology and Stem Cell Biology at Ulm University in Germany. The title of my talk is Treatment Response Prediction and Capturing Tumor Evolution Using Pancreatic Cancer-Derived Organoids. So before I go over the details of my presentation today, I would like to introduce to you pancreatic ductal adenocarcinoma and why it is such a challenging disease. It is the most frequent. Pancreatic malignancy. It is facing a high rate of heterogeneity, a high rate of mutational complexity also, and there is a lot of intrinsic therapeutic resistance. When you have a look to the overall 5-year survival rate, it is just above 10%. And at the moment, pancreatic cancer is the first cause of death, cancer-related death in Europe. And when you look to the predictions, it will going to be second ranked up to the year 2030. The chemotherapy, the therapeutic opportunities are mostly limited to conventional chemotherapy, and we have just 2 regimens, which I will explain later to you during my talk. And just a few patients qualify for curative surgery, and just about 2% qualify for a kind of a targeted therapeutic approach. So the question is now, will we ever find a so-called one-size-fits-all medicine in pancreatic cancer, where we really have a good efficacy with few side effects for all our patients? And at the moment, it is the belief of the community that at the end of the day, we have to treat basically each and every pancreatic cancer as an individual disease. So we really have to personalize the medicine, the pancreatic cancer medicine, and to approach the heterogeneity, the intrinsic resistance, and thereby spare toxicity to our patients with the applied chemo. So why is it now? So why is pancreatic cancer such a hard to treat cancer? And I think this is best explained when we have a look for its molecular makeup. And these are the most recent data from the Cancer Genome Atlas summarized in 2017. So this is still state of the art. And when you have a look here, there are just a few core driver mutations. And here the most relevant player are activating oncogenic KRAS mutations. So as you can see, most of the PDACs are mutated in KRAS, but they also, a relevant proportion loses the tumor suppressor gene p53, but also CDKN2A and SMAD4. And besides this core set of driver mutations, you can see there are a variety of passenger mutations. And all these passenger mutations, and here we also have to mention, for example, DNA damage response genes. Here the most famous player is BRCA, and here we have the only approved targeted therapy in pancreatic cancer. So this passenger mutations refine the heterogeneity of the disease, and thereby we really, really need to approach a personalized medicine approach in pancreatic cancer. But the question is now, when the genomics just in rare cases educate treatment, the question is now, can we come up with a phenotyping approach instead of a genotyping approach to personalize PDAC medicine? And here organoids might be a good tool to approach pancreatic cancer from a predictive point, a predictive point of view. But what are organoids now? Organoids are a three-dimensional culture format, and they recapitulate an origin or a cancer in structure and function. And when we have a look to the culture conditions, the most important thing is that we culture them three-dimensionally in a kind of a niche. We usually use matrigel or other hydrogels resembling the niche of the pancreas. And usually we supplement the media with certain growth factors, and organoids can be grown from cancer, from tissue, and from tissues then mostly from stem or progenitor cells. But we can also generate organoids from human or mouse pluripotent stem cells to cultivate them in this 3D format. Organoids have really pioneered medicine, particularly also cancer medicine, and therefore Nature Method has ranked organoids already in the year 2017 as the method of the year. So what my lab is doing now, and how we are approaching pancreatic cancer, is basically from two sides. This is just summarized in a recent review which we published in Trends in Cancer. So we are approaching pancreatic cancer either by differentiating human iPS cells from patients or embryonic stem cells, which are CRISPR-Cas edited for certain tumor suppressor genes. We differentiate them to either ductal or acinar cells, and then study the early stages of pancreatic cancer with this model system. However, on the other side, we are also using PDOs, so basically pancreatic cancer organoids derived from end-stage cancer, either derived from the metastasis, but also from the primary tumors. And here we can do phenotyping, pharmacotyping, and thereby eventually use them for drug response prediction. What I'm not going to touch, but is really relevant and worth to mention at this stage, is that all these organoids are lacking at the moment the tumor microenvironment, and we really need to approach and model the tumor microenvironment to properly tackle pancreatic cancer, probably in the next decades. So What are the take rates in general for organoids? This is just a review from 2018. You can see that the efficiency for cultivating the organoids is fairly high, and in pancreatic cancer, the take rate is around 8%. But here I really would like to draw your attention that efficacy and the definition of efficacy really varies amongst the various studies. And from my point of view, I would really mention here, it is important to see whether it's not only able to cultivate them, but it's also possible that you define take rate as the possibility to really perform a pharmacotyping with the derived organoids. So the question is now, what can we do with organoids? They are reliable in fast generation across various tumor entities. They model a whole spectrum of various cancer types. They also approach tumor subtypes. And we have invested quite some efforts to really use them for drug response prediction in pancreatic cancer. And this is the story what I would like to wrap up with you over the next couple of slides. So we started a couple of years ago to set up an organoid pharmacotyping pipeline. And here we used xenografted tumor material. So patient tumor pieces have been expanded or grown in mouse xenografts. So we took the pieces isolated from the frozen material organoids. We established a pharmacotyping pipeline. And then we treated the mice, the tumor pieces, with the various drugs. So the 5 relevant chemotherapy drugs employed in clinical routine. So this is gemcitabine, paclitaxel, irinotecan, 5-FU, and oxaliplatin. And then we compared the drug sensitivity of the xenograft in the mice and of the organoids in the drug sensitivity assay. And this is just a summary of this published data. There was a high match of the xenograft and of the organoids in the response to the various drugs. We also used the organoids to derive biomarkers here as shown, as summarized in this regimen or in this scheme, using deriving extracellular vesicles for biomarker derivation. But as I told you when we started, this was all xenografted human tumor pieces. So we really wanted to approach organoid medicine from a real to implement it in clinical routine. So we started to sample primary human tissue and wanted to apply this in a prospective kind of a feasibility trial and really test the organoids for their predictive capacity. So we isolated either primary tissue or metastatic tissue, set up the cultures, we defined the pharmacotyping protocol, phenotypings, we did phenotyping of the organoids and also biobanking. And then we prospectively sampled the organoids, tested them, and then the patients, we compared the patient response after the first staging with the response of the organoids in vitro to really see whether we can make a prediction in clinical routine. So let me just summarize these data. At the very beginning, we did a phenotyping of the organoids, the PDOs versus the primary tissue. You see there is a fairly good concordance. There is a trend to switch more to the basal-like subtype, but overall the subtype remained pretty stable. We also benchmarked our pharmacotyping efficacy, and I just mentioned this before. So we really believe that it's important to test the organoids for the pharmacotyping efficacy and not only whether they go for 1 or 2 passages. So here we had a derivation efficacy of around 63%, and you can clearly see that this is obviously dropping and differing whether you have a treatment naive organoid or a pre-treated organoid or an organoid which is coming from the primary tissue. We also set up a pharmacotyping method, and these are the classic pharmacotyping curves which we get here used for shown for gemcitabine. And we used a mathematical method to the AUC. It's called Jenk's natural breaks to define high responders, intermediate or low responders by cultivating a score to the 2 main regimens. The question is now, can we use the readout? And as I told you, we compared now the drug response of the organoids in this study with the first staging. And here in this green box, you can see the prediction accuracy, which was for first line more than 90%, and for the second line in just in a small sample size around 80%. However, it strongly dropped when we tested pre-treated organoids. And when then the oncologist, by chance, so the organoids did not inform or educate the decision-making of the oncologist, but we could retrospectively compare in case the oncologist has chosen the regimen which was predicted to be responsive based on the organoids, the survival was really better, which is all in these patients, which is also shown here in these swimmer plots, as you can see on the left. So these data really were the basis for establishing at Ulm University the first core facility for organoid medicine, which was just tailored to generate a living biobank to derive organoids not only from pancreatic cancer, but also from various tumor entities. We standardizedly biobank the samples. We have a pharmacotyping protocol of them, and then the clinical scientists can make retrospectively, but also we are conducting clinical trials for the clinical response prediction. We offer method training, scientific services, and we have staffed the core facility with technicians and the technical head, as here shown for Jessica Lindenmeyer. So now the launch point of the core facility is 2 years ago, and I can now present to you real life data from after 2 years of a standardized operation of such a core facility, which is just summarized in this workflow. And what we are doing here, we are sampling resectable tumors, unresectable. We are doing rebiopsies on recurrence, but also upon progression to really capture tumor evolution. We biobank them, pharmacotype them. We do also molecular profiling, including single cell sequencing, and we do the classical drug response prediction. And we also used these data to operate and launch the first clinical trials. So what are now the real life data? Meanwhile, we have sampled more than 100 tumor samples from pancreatic cancer. The successful establishing rate was 86. There's some organoid lines still pending, and almost 80 went into the analysis. And I just would like to draw attention to these 51 cases. These are the patients which did not reach the first staging. This just underpins how deadly this disease is. So at the end of the day, we just have 28 patient samples with clinical correlation data. But another lesson, before I show you now these real life data, I would like to mention another hurdle which we were facing in our initial feasibility trial, and this was timing. Timing in terms of how long do we need to produce pharmacotyping data with the organoids in clinical routine. So at the beginning, we were slow, so we changed now when launching the core facility to a robot-based drug screening. We compared the robot with manual assembly. You can clearly see that the drug response category did not flip. And the most important thing is what we have now with the robot, we can increase our capacity, so we can test pharma drugs in parallel. And most importantly, we cut down the time to provide the pharmacotyping data to around 30 days, which is really feasible in clinical routine. And thereby we have we generated the basis to really, in theory, employ PDO testing in a clinical trial setup for the future. We also can do now, so what can we do? We can also do phenotype-based reverse genotyping. So I would like just to draw your attention to a drug response curve for olaparib. And here, this is a longitudinal sample. First biopsy, the patient was clearly responsive. We genotyped the patient here. He or she had a CHEK2 mutation. And after treatment with olaparib, the patient became resistant. We did another biopsy, and you can clearly see that the resistance is clearly mimicked in a response category shift in the PDOs, which is also mimicked in cisplatin, which is known for DNA damage response genes, that they are also not only sensitive for PARP inhibitors, but also for plating derivatives. So how our data now, we have a success rate of 61%. Again, we have differences in treatment naive versus pre-treated cases, but also in primary versus treatment naive patients. And we also saw a great difference in the assemblage of the material, either when we use ultrasound-guided pre-cut biopsy, EOS-guided, but also from surgical specimens. But the question is now, how is it now performing? How are the organoids performing in real life, operating from a standardized core facility? So these are, again, the percentage of change in the target lesions according to the RECIST criteria. And I would like to just draw your attention to this category, the response prediction to the applied regimens, and you can clearly see there is a high match, and the overall prediction efficacy now in real life is 80%. So it dropped from the initial feasibility trial, but in clinical routine, we can clearly predict the response of the patient by 80%. And the interesting thing is that we can better predict the responsiveness of the tumor compared to the unresponsiveness, and we are studying this now in more detail. And again, when the oncologist has chosen the right regimen, this is mirrored in the benefit in the survival rate. I told you we also did longitudinal sampling during therapy, so we did a second organoid line up in tumor progression. This is just one set of 7 patients where we did whole exome sequencing. You can clearly see, without going into the detail, that the mutational landscape is changing. But what I would like to draw your attention is this plot here, where we use the computing tool to calculate the clonality of the organoids before and after treatment. You can clearly see that the clonality, so the heterogeneity, is going down to where we have a clear selection of mutant or probably of mutant clones, which also present with a different mutational makeup. So to sum up what I have shown you today, PDO-based pharmacotyping is feasible with a reasonable time frame. There is a positive correlation with the drug sensitivity prediction and the therapeutic response of the patients. It remains true in daily clinical routine. This is also directly translates to a survival benefit of the patients. And now we believe we are in the position to really, and this is the most important thing for today, and I think also a take-home message, we have to probe now organoids in clinical standardized trials, and we are now just starting to get the approval of the Unite Punk trial, a prospective phase 1 proof of concept trial on resectable pancreatic cancer. And this is really important to test, because otherwise, without clinical trials, we cannot really say whether organoids are really a robust preclinical tool to decipher and response prediction. And we can also, and to see whether they are suitable also for further molecular analysis. And this is my last slide. I would like to thank the people who have done the work, and this is a collaborative study with the Department of Internal Medicine and the Institute of Medical Systems Biology. I've underlined the important people who have contributed, and I thank you for your attention.
Thank you so much for joining us today, and thank you for Sartorius for sponsoring this, and for Science for putting this on. My name is Connie Liebachen. I'm one of the co-founders of STEM Farm, which is a startup based in Madison, Wisconsin, and we are focused on developing a model of neuroinflammation that is built from iPSC cells, and importantly, we can incorporate microglia. So I'm very excited to present our data to you today. So the challenge that we're facing is that there are very few effective treatments for neurological disorders. However, it's a big economic as well as personal problem. So 1 in 6 people worldwide have a neurological disorder. The economic impact is incredible. In the United States alone, it's $800 billion is the impact of neurological diseases to our economy. And clinical failure rates of these for these diseases is incredibly high. So really, we need better physiological in vitro models for CNS drug discovery. Human models, including organoids, are gaining adoption to improve clinical translation. So we really want to move from curing animals, but not patients, to curing patients based on predictive human disease models. So cell lines in animal models fail to predict clinical outcomes, and greater than 40% of clinical failures are due to a lack of clinical efficacy. And we all know that late-stage failures cost hundreds of millions, if not billions of dollars. So pharma companies are adopting in vitro 3D models for drug discovery, and organoids can model many aspects of neurological diseases. So our organoids are built in a different manner than a traditional kind of organoid, and it's based on our proprietary synthetic hydrogels. So our academic co-founder is Bill Murphy at the University of Wisconsin-Madison. He has developed synthetic hydrogels. These are PEG-based hydrogels where we can modulate the mechanical properties as well as the cell adhesion properties and the degradability of the hydrogels over time. So we build these in a successive manner wherein we start with the organoid, or we start with the hydrogel in a 96-well plate. We add unspecified but high quality neural precursor cells, culture those for a period of time in proprietary media, add endothelial cells and MSCs, and finally we can add microglia to the culture. All of this is done in the context of 21 days. And so at the end of 21 days, we have a planar kind of pancake-shaped morphology neural organoid, as is demonstrated here in the micrograph, that contains microglia throughout. So these are about 10 cells thick. This means that we don't have a necrotic core. We have very good access for dosing regimens, and it's a great way that we can image as well. And I'll show you some images later on in the presentation. So the context of use is very important when we're talking about organoids. Organoids cannot fulfill every criteria for an organ. And so it's really important to think, what can these organoids be used for? And the context of use for our organoids really are around modeling disease, discovering new targets, doing phenotypic drug screens, and evaluating neurotoxicity. So this is a figure taken from a recent publication that we had in collaboration with the Murphy Lab, recently published in the Journal of Tissue Engineering. And here what we have done is we've looked at a transcriptional comparison between the organoids built in the stem farm manner versus traditional organoids. And you can see on the image, right, human iPSCs are generated in both cases. The organoids then, in a traditional manner, are matrigel suspension, differentiated in that, kept in suspension, and they form a round kind of structure that might have a necrotic core in the center. Whereas our organoids are built on these PEG hydrogel surfaces, we develop these planar neural organoids. So in this case, the same cell types, the same iPSC cells were used to generate organoids for this. And then we did bulk RNA sequencing. So you can see on the far right-hand side, this is gene ontology analysis, looking at the differential gene expression between these organoids. And importantly, I'll bring you right to the bottom of the graph here. And what we have plotted here is the number of genes in each gene ontology set that are affected in that gene ontology set, but importantly also the p-value. And you can see that these are very, very statistically significant changes. And the one that is the most statistically significant is actually the extracellular matrix organization. And so the hypothesis here is that the stem farm synthetic hydrogel enables cells to generate their own ECM, better modeling that of the brain microenvironment. So rather than having these cells that are surrounded by matrigel, they're generating their own and then differentiating. You can also see that there are some expected changes because we're adding microglia. We see a lot of changes in the innate immune response. Because we're adding vascular endothelial cells, we see development of angiogenesis and such. But we also see some more maturation around neurotransmitter secretion, postsynaptic density, and such. And so we're really excited that our model is able to recapitulate both the ECM to generate a supportive environment, but also maturing neurons. So we've done single-cell transcriptional analysis of our organoids to demonstrate the diversity of cell types that are present. So the t-SNE plot on the left demonstrates the different clusters that we've generated. You can see that we are generating from the IPS, from the NPCs, we get radioglial cells, proliferating IPCs, early and late neurons, and astrocytes. And then the other cells that we're adding, the microglia, endothelial cells, and MSCs, are all present. So we identified these clusters through enriched gene ontology terms, as well as looking at key marker genes from each cell type. Our organoids are highly reproducible. So this, again, is a single cell experiment on the left, where we've taken 3 replicate organoids and put them through a 10x genomics single cell protocol. You can see that the t-SNE plots are very, very similar, and the Spearman rank correlation coefficients below them match very, very well. Bulk RNA sequencing is also very consistent. So on the right-hand side is data from a bulk RNA-seq experiment, wherein we're looking at 4 replicates of either control or LPS-stimulated organoids. And you can see that the control genes in the black are very consistent between the groups. We have markers for endothelial cells, microglia, astrocytes, NPCs, and maturity markers. And importantly, upon LPS stimulation, we see an increase in IAF1, the IBA1 protein in the microglia, and a decrease in SOX1 in the NPCs, so demonstrating some specificity. The tightness of the bulk data has been very useful for us to look at treatment paradigms and do bulk sequencing and look at gene ontology analysis with the organoids. But one of the highlights of our organoids is that we can incorporate the microglia into them. And so really the question is can we assess the diversity of microglia phenotypes in our organoids? People like to think of microglia. So microglia are the macrophages of your brain, essentially. Like to think of them as either activated or inactivated. But in reality, it's more of a spectrum of phenotypes. And often these phenotypes are built based on feedback loops from the microglia themselves. And so it's important to think of the microglia phenotypes in this manner. The microglia that are incorporated into our organoids display a ramified morphology and develop an in vivo gene signature data set. So there was a nice meta-analysis done by Patir et al. In 2019 that was published in the references below, where they identified 249 gene set that identified in vivo microglia. And in fact, we have a very nice match with from the single cell data with the microglia of this gene set. You can see that we have very specific expression of expected markers, and the micrographs in the middle, show that the microglia are very ramified. This is a confocal image with a maximum intensity projection, so kind of looking at all of the projections at once. And they're interacting both with the neurons as well as the astrocytes in that population. So really the questions we've been asking is, can we assess the diversity of the microglia phenotypes in our organoids? And we've done this by generating organoids and then treating them with a variety of biologics or compounds to develop an anti-inflammatory phenotype, a pro-inflammatory phenotype, depleting the microglia either with the CSF1R specific PLX3397 or not adding the microglia at all. And then also looked at neurotoxicity. And the readouts that we're building are transcriptional, protein-based immunoassays, looking at the morphology of the microglia and the neurons, and then basic cell health and some functionality work. So we can use multiplexed immunoassays of supernanes to utilize, can be utilized to assess the organoid responses. So in this experiment, we've taken either control organoids or organoids that have been treated with 50 nanograms per mil of LPS and interferon gamma, and looked at a Luminex-based multiplexed assay. And you can see that we very nicely can pick up a neuroinflammatory response upon that treatment. So here on the left-hand side are analytes that are in response are increasing in the supernatant in response to LPS plus interferon treatment. And then on the very right side, we've put some controls which aren't, just to demonstrate that this is a specific phenotype. Now, some of these analytes are microglia specific and some are not. And we can analyze that either by looking back at our single cell data to look at where the response is coming from, or we can generate organoids that don't contain the microglia and look at those responses. We've been very interested in applying this preclinical model to how it translates to clinical applications as well. And so we want to continue to look at biomarker assessment. And so this is an experiment that we did in collaboration with collaborators at Firesite Therapeutics, Yanling Wang and John Levinson. And we generated the organoids and the treatment samples, and then they performed a ProCarta analysis with neurofilament heavy, UCHL1, or S100 beta. And this demonstrates a very nice specific response to rotenone with these treatments. So on the left-hand side, we have neurofilament heavy, which is released with ethanol treatment, so 20% ethanol, essentially no effect with rotenone, and released with colchicine. The UCHL1, which is a ubiquitin ligase family protein that is neuron specific, has a very similar response, whereas the S100 beta has a very high rotenone response, and that is known to be an astrocyte specific marker and is released specifically upon rotenone treatment. So it's very exciting to be able to use this in vitro organoid model to look at clinically relevant biomarkers. We've also done a lot of work assessing the microglia phenotype in the context of our organoids. So here we're measuring sphericity and volumes as morphological metrics. And as the microglia are not very sphere-like, they're very ramified on the far left-hand side, have a sphericity measurement of about 0.27, and a microglia that would be very, very activated would have a sphericity measurement more sphere-like, around 0.67. So we can measure individual microglia responses through imaging analysis. On the bottom, we have control organoids that demonstrate that ramified phenotype. So in this case, we're using GFP-tagged microglia and incorporating them in. You can see that they are organized in the YZ plane throughout the organoid. When we treat with interferon gamma, the microglia dramatically change shape. They look more motile. There's some clustering of the microglia, as well as the astrocytes. If you look at the YZ plane, the astrocytes are also becoming activated and are actually penetrating into the hydrogel on the far right of that image. LPS plus interferon has a different response in that the microglia are becoming more amoeboid. There's some thinning of the hydrogel or of the organoid in all, and we're also seeing that astrocyte response. We've used these measurements to understand better the integration of the microglia into our organoid. So if you remember, the microglia are coming into the system last, but yet we're getting really nice incorporation. So we wanted to watch that over time, and we could use the sphericity measurement to do this. So here we have taken live cell images of one single frame of an organoid as it's developing. So day 1, post microglia addition, all the way to day 8. And if you look on the graph on the right, you can see that the sphericity measurements change dramatically between day 2 and day 3, whereas that's when we see the microglia take on this more ramified homeostatic phenotype, and that is maintained throughout day 8. So we have been fortunate to receive funding through several SBIR grants, and we've been funded by NIEHS to use this as a developmental neurotoxicity platform. So we're looking at toxicity in the context of this 3D human biology. So in our phase 1 work, we took some known neurotoxins, including chlorpyrifos, lead acetate, valproic acid, and then a control compound for the microglia, PLX3397, treated the organoids for those, and then did bulk RNA sequencing to look at transcriptional outputs. On the right-hand side is a heat map of gene ontology sets that are regulated in response to these treatments. So you can see that chlorpyrifos is dramatically downregulating neurogenesis, metabolism, cell proliferation, and ECM and cell motility, whereas valproic acid is actually having an upregulation of the ECM, but a significant downregulation of inflammation markers, and that is something that's known. And valproic acid, while used as a seizurogenic compound, is also being explored to attenuate microglia responses from activated microglia. PLX3397 has a very specific response in the downregulation of the microglia-specific genes, and lead acetate then has a mixed, a decrease in ECM cell motility, and that is expected in the literature as well. So these transcriptional phenotypes that we're observing in the system are consistent with the mechanisms of action that have been described for these compounds, and we're continue to pursue this developmental neurotoxicity. Now for a case study around neurodegeneration utilizing these organoids. So I start with an example of microglia in monoculture, and you can see those microglia on the far left-hand side, where we have control, or we have added a cipher-labeled A-beta peptide. So the cipher labeling will cause when that peptide is taken into the cell and is in the lysosomal compartment, it will become fluorogenic. And so we can measure the phagocytosis of those A-beta peptides. And you can see on the figure moving to the left that we see a dramatic increase in fluorescence intensity when the A-beta is phagocytosed. We do not, however, in 2D, in this monoculture, observe an increase, and this is a qPCR readout, of TNF-alpha, IL-1 alpha, IL-1 beta, or Chi3L1. In the control, LPS plus interferon, we get a robust response, but not in the presence of the A-beta oligomers. So we asked then, is that the same or a different response in the context of the organoids? So when we do the same experiment in the context of the organoids and might measure the microglia response, we can get an inflammatory response to A-beta. So on the left-hand side here, we have the organoids, either control or A-beta. The A-beta does not affect the morphology overall of the organoids, but you can see then in panel B, where the microglia are labeled in magenta, and then the amyloid aggregates are labeled with methoxyxO4. So you can see on the far right of that pictograph that the microglia are interacting with those A-beta oligomer aggregates, and that we can get a detectable response to TNF-alpha and Chi3L1. It's not a huge response, and that's something that we're working on, is seeing if we can see an increase in that response. But it's definitely statistically relevant and significant. So the next experiment that we did was we built organoids that either contained or didn't contain microglia to ask if these were specific responses to the microglia. And we added the A-beta oligomers. So you can see that without microglia present, we have no IBA1 signal, which is expected. And with treatment of the A-beta oligomers, we get a response, an increase in signal, although not statistically significant at this point, of IBA1. We do get a statistically significant response of IL-1 alpha as well, and TNF-alpha and Chi3L1 in the presence of the A-beta oligomers. The pictograph on the right shows that in the context of the organoid, we're getting phagocytosis of the A-beta oligomers as well. So here we have done staining for IBA1 in green, or CD68, which is upregulated when microglia are phagocytosing. And you can see that we see co-localization of the IBA1 and CD68 upon treatment with the A-beta oligomers. We then asked if we can expand this to general neurodegeneration, and would we get a response to apoptotic neurons. So we took iPSC-derived GABA neurons. We treated them with UV exposure and did a pulsed controlled UV exposure 5 times. and then grew the cells for additional 24 hours, wherein they became apoptotic. We then harvested them and added them to the organoid to look for a response. And you can see that we're getting a differential response between the apoptotic neurons and the A beta. So I'll talk you through that a bit. So we see an increase in IBA1 mRNA levels in both cases. We see TNF-alpha response to A beta oligomers, but not a specific response to the apoptotic neurons. However, we see upregulation of CD68. So they are phagocytosing these apoptotic neurons or pieces of the apoptotic neurons. With the A beta, we see an upregulation of C1qa, which is not statistically significant with the apoptotic neurons, as well as CLEC7. And interestingly, we get upregulation of the P2RY12 with the A beta as well as TMEM119. And on the right-hand side, you can see that increase in the IBA1 and at the protein level as well. So on the top, we have IBA1 controls, and then when we express the when we add the apoptotic neurons, we're getting much more IBA1 signal. So that's the data that I have to present today. In conclusion, STEM Farm is driving insights with our neural organoid, insights into CNS biology. We're doing this by having unprecedented access to relevant human neurobiology, our biomaterials that mimic the physiological environment. We've done a lot of work to validate this organoid platform that incorporates microglia and know that it's a repeatable, robust format, and it's ideal for studying neuroinflammation in vitro. And we are open to drug discovery partnerships. So please contact us if you're interested. We're doing contract work as well as collaborative work, and happy to discuss those opportunities. I'd like to thank the STEM Farm team. It's a great team to work with, and Bill Murphy and his lab, in particular, Joy Deb, who was the lead author on the manuscript that I shared, our collaborators at Firesight, as well as collaborators at Illumina. We were an Illumina accelerator company, and just benefited greatly from that experience in the sequencing that we could do, as well as just getting our bioinformatics pipelines set up and worked. The imaging is done through the University of Wisconsin Optical Core. Our IP is from WRF through the University of Wisconsin, as well as developing our own IP. And then we have received great support from NIH, funding through EHS, NINDS, and NIA, and NCI to develop this platform.